Firstly, I'd like to say thank you to the Danish Royal Society for giving me the opportunity to speak today. It's a great privilege, and of course, thank you to L'Oreal Foundation and UNESCO for the prize. Today, I'm going to talk to you about some of my work in the area of stimuli-responsive smart materials. And let me say from the beginning that when chemists talk about smart materials, we don't mean that they're smart because we designed them well. We mean that they're smart because they have some sort of intelligence of their own. So they have the ability to respond to their environment and adapt to changes in their environment. Now, when I talk about a stimuli-responsive system, I mean a system where there's an input, which leads to an output that's not a direct consequence of that input. In between, there's some sort of machinery that converts the input to the output. And this is best explained, perhaps, with some examples. So let's take quite a simple one. This is probably where we teach children about stimuli-responsive systems. In this system, you press on the pink button, and to the surprise of the child, suddenly the dinosaur pops up. And it's a surprise because the input was to press down on the button, and the consequence of that, you would expect, is that the button simply goes down. If you wanted to open up the dinosaur, you'd take it in two hands and pull it apart. That would be the anticipated response. But instead here, we have an input, and then an unexpected output that's the consequence of some mechanics in the background. So inside the blue box, there's some machinery which converts our input into a different output. Another example would be turning on a light. All we do is press the button, and of course, then the switch does move. But as a consequence, on the other side of the room, the light turns on. And in this case, there's some electronics in the background, and an electrical process converting the input to the output. This also happens in biological systems. So if you touch something hot, the typical response to that stimuli is to step back, ow. In this case, there's quite complex biological machinery in the background. Your hand senses the heat that conveys a message to the brain, which tells you to step back and leads to the shouting. So these are three examples that use mechanics and electronics and biological systems to convert an input to an output. Now, what my research is about is trying to build chemical systems where we have molecules which do basically the same thing. And in this case, what we're looking at is molecules that can change their shape or their structure so that you get a different output depending on the input that you give it, the system. So to outline how this could be useful and what is, why we do this research, I'll move straight to an example of how this could be applied. And if we think about how many drugs are administered, mostly they're taken orally or injected into a vein, and then they have to transport themselves around the body to the location of the infection or the illness. In many cases, therefore, it's desirable to put this drug inside a molecular capsule, which protects it and helps it to transport it to the location it's needed. Now, what would be even better is if when it got to the location it was needed in the body, we could have some sort of stimuli which caused the capsule to break apart and release the drug. The stimuli could be physical or chemical or biological, so it could be the application of light or heat change or pH change or an enzyme. But the consequence is the same. The drug, after it's been delivered to the location it's needed, is then released and starts to act. If we put this in the context of how cancer, for example, is treated today, typically it's treated with chemotherapy, and people tend to get very sick from the drugs, which are acting on the tumor, but they also act on many of the healthy cells in the body. So this sort of technology would make it possible to design better drugs and better delivery systems so that people don't get so sick from the medicines they're taking. Now, if we think about how the work that needs to be done to develop these sorts of stimuli-responsive medicines, it takes the collaboration of lots of different areas of science. It's not just chemists. So the chemists maybe synthesize the drugs and the materials, but then we need to collaborate with material scientists who can understand how and study the stability of the materials and the degradability. Biologists and biochemists who can study the actual effects of the drugs on the body, and of course, doctors who would study how best to administer the drugs, how best to administer the stimuli to make it work effectively. But I am a chemist, so I'm looking at the first step in the process, which is how do we actually make molecules that are stimuli responsive. So the role of chemists in this area would be to design and synthesize the drugs, but also the carrier molecules, and look at ways to release the drugs from the capsules, look at new ways to assemble molecules around capsules, and then, of course, consider all the practical implications, like that it needs to be degradable, that it needs to be biocompatible. And so this brings me to the areas I'm going to talk about today. And to start with, I'll talk briefly about how can we make molecules that react to different types of stimuli, and then how we can get molecules to spontaneously self-assemble to build materials that themselves are stimuli responsive. And then I'll move on to materials based on carbohydrates, which is the project that I'm working on now and for which I'll be using the prize money. And there I'll talk a little bit about how I got interested in carbohydrates as well. So as I've said, in a chemical stimuli-responsive system, we can have various inputs, various outputs, and in between the molecules will rearrange themselves in some way. And the inputs could be a temperature change, pH change, signaling molecules, enzymes, catalysts. There's lots of different possibilities there. The sorts of outputs we'll be looking at is something like guest release, which is the example of the drug delivery system. But also subsequent reactions and optical responses, which would be relevant for making sensors. And then also physical changes in the material. So if you have a polymer, which it might be like a plastic, and then if you apply a different stimulus, it goes from being a hard material to a soft material. So there can also be macroscopic changes that we can see in our hands. So let's take an example where we use light. This is a molecule called diazobenzene, and it is a special molecule because it has the ability to switch its shape from being a linear structure to a bent structure. If you shine light on the molecule, a UV light, it spontaneously rearranges. It doesn't have to change any of the atoms around, it just rearranges the orientation of these atoms. And then we have a different structure, and that, of course, means that we can get a different function. Turns out this sort of mechanism is actually what enables us to see. So this molecule here is called retinal, and as you can see, it has a bent structure. It's located on the back of the eye in the retina. So when light enters the eye, it causes this molecule to rearrange and go from a bent structure to a linear structure. And this affects how the molecule binds to protein opsin, and that in turn initiates a cascade of biochemical reactions which results in the brain recognizing the image. So in fact, many of the materials that chemists design are very much inspired by biological systems. And here's an example where we use pH. We can use pH change or signaling molecules to activate the system. So this is a molecule that we call a rotaxane, which is basically like a donut on a piece of string with two stoppers at the end so that it can't fall off. And in this example, the donut has two options as to where it can sit. It can sit around this red part of the molecule or about the green part of the molecule. And we can cause it to switch from one position to another position by changing the acidity of the solution. So under basic conditions, it will sit around the red part of the molecule. And then if you add acid, it switches its position. In this particular example, the green part of the molecule is actually catalytic and acts as an organic catalyst. So what you have here is a system where you can switch on and off a catalyst by changing whether it's in a base or in an acid. This sort of changing of structure based on a signaling molecule is also very important in biological systems, and in fact how our brains work. So when neurological messages are transported around the brain, this happens via a process where ions move between different cells. And they're typically, you have a protein that's located inside a cell membrane, and then there's a ligand or a signaling molecule which binds to the protein, causes it to change its shape so that it opens up, that allows the ions to flow through. So you have an input as the stimuli, the ligand here, the output is that it's basically like a gate. It can open and it can close. Now in both of these examples, we had a single molecule which changed its shape as a consequence of the stimuli. But another way of making these sorts of systems is to take a single molecule that can assemble itself in different ways and then try to influence how it self-assembles. So if we imagine a triangular-shaped molecule that has the ability to stick to itself, it can do so and make a linear structure like this, or it could make a cyclic structure. And if we're able to influence which one of these structures it chooses, then we have a way of influencing the properties of the system. So if I can shine light and switch from here to here, I haven't actually changed the molecule, but I've changed how the molecules interact with one another. So this would be called a supramolecular system, and this is the area that I'm most interested in. In fact, this idea of molecules spontaneously coming together to form responsive structures is something that humans have been exploiting for hundreds of years. So let me explain how soap works. Soaps basically consist of molecules that have two sections, what we call a hydrophilic head group, which means section that likes water, and a hydrophobic tail, so something that hates water and likes oils. Now, if you put this into water, it spontaneously forms into spherical structures, which we call micelles, where it puts all of the tails that don't like the water in towards the center, and all of the head groups that do like the water out towards the surface. So when you're doing the washing up and you have oil in the saucepan and on the plates and you try to just wash it with water, it's not going to work because oil and water don't mix together. But if you add a soap, then the oil from the oil on the plates can enter inside the center of these micelles, thereby getting away from the water, and then it dissolves and can be washed down the sink. So this is a process that we're all very familiar with, and it's actually the basis for some of the chemistry I'm doing. What would be interesting, of course, is if we could intentionally put a molecule inside these micelles, and then also have a way of releasing it. Because then you have, for example, a drug delivery system, or you have a delivery system which can transport species around. So let me take an example from some work I've been doing recently. Here we have what's called an Anthro File. So this is a molecule which has one section that likes water, one section that doesn't like water. And in this case, the head group likes the water because it has these negative charges. If you put it into a solution of water, it spontaneously forms micelles, these spherical structures. In this case, there's 3 negative charges, and like charges repel each other. So actually, it doesn't work so well, so we add another component, which is this molecule called spermine, which has got 4 positive charges. So this basically acts like a glue. Positive charges and negative charges attract, and then this stabilizes the structure. So now we have a more stable capsule. But it's not enough to be able to build the capsules. We need to be able to make them break apart and reassemble and control all of these processes. So I've been working on a system to try to do exactly that, break it apart, put it back together. And let me take you through it step by step. First step involves breaking the micelle apart by trapping one of the components inside of another molecule. So using a molecular encapsulation. Now what happens here is that we have a receptor molecule which is made up of 7 glucose residues, and the inside of this is hydrophobic, the outside is hydrophilic. So what this means is if you have a molecule that doesn't like water, it tends to insert inside this macrocycle. And that's exactly what happens here. So we have a competition, and if we add enough of this beta-cyclodextrin, then you can break the micelle apart by trapping one of the components inside a box. Of course, we need to be able to reverse this process. The way we do that is to add an enzyme. In this case, it's called alpha amylase, and it's an enzyme which takes this glucose macrocycle and breaks it down into smaller pieces. So if you add the enzyme amylase, you can then switch back to the micelle because we've now destroyed the capsule that we had trapped the molecule in. We can do this another way. Instead of trapping the Anthro File, we can trap the spermine molecule. And here we use a receptor that has 8 negative charges, and they interact with the 4 positive charges on the spermine, thereby removing it from the system and releasing the Anthro File. we can then reverse the process again by breaking down this receptor. We do that in an electrochemical process where we basically transfer some electrons from this compound to this one, which causes it to break apart, and now we've got our micelles back again. So we have a system where we can create micelles, pull them apart, put them back together again, and obviously the next step would be to put something inside. Put something inside and see if we can release it in a controlled manner. That's something I'm working on. But you might be wondering, how do I even know that these systems are really doing what they're doing? I mean, it looks quite nice on paper. But now we can't, all of this is happening in water. So we can't look at it under a microscope because the molecules are too small. We can't do X-ray crystallography and look at the 3D molecular structure because we're not interested in what the solid looks like. We're interested in what's actually happening in the water. So we have to use a variety of indirect techniques. And one that is particularly relevant in my work is called nuclear magnetic resonance spectroscopy, NMR spectroscopy. Basically, we take the sample as a solution, put it in one of these small glass tubes, insert it inside a very big magnet, and then use radio wave pulses to excite the sample, and we get a response whereby we get a different response for all the different hydrogen atoms on the molecule, depending on their chemical environment. We get a spectrum that looks something like this. Now, if we consider the molecule I've been talking about, it has 6 hydrogen atoms on this head group, and we get 6 signals down this end of the spectrum. Meanwhile, the hydrogens on the tail are in this part of the spectrum, and we can assign each of these and interpret the structure of the species. Now, when it turns into a micelle, the chemical environment around these hydrogens is now very different. So this now is the spectrum of the micelle, and you can see there's still signals for each of the hydrogens, but they've now broadened and they've moved around, and we can follow this process. If now we add one of our molecular encapsulation boxes, which takes away the micelle and releases the Anthro, then we see that these blue peaks here are exactly the same as the ones we saw up the top. So that means we really do have this molecule now free, floating around in solution. So we can actually follow what's going on by doing it step by step and following the process. Another important technique is fluorescence spectroscopy. What I didn't mention is that this molecule here is actually fluorescent. But when it forms a micelle, it stops being fluorescent. So if you shine UV light on it, it looks blue, and when you form a micelle, it gives no signal at all. This means you can actually just tell by looking at the sample if it's a micelle or not. But if you do it more precisely and use a fluorescence spectrometer, then you can follow the process of assembly and disassembly. So on the y-axis here, we have the fluorescence emission, so the strength of the light being let out. And moving from left to right, here we have the free Anthro file. Here it's formed a micelle because we added spermine. Now we add SPOX, trap the spermine, and release the Anthro file, so the fluorescent signal comes back up again. Now if we reduce the molecule and break apart the capsule here, we reform the micelle, and you can see that the fluorescent signal disappears. So you can follow step by step the process that's going on. So this brings me now to the final section of my talk, which is to talk about enzyme stimuli-responsive systems that are based on carbohydrates. Most of the time when we talk about carbohydrates, we're talking about carbohydrates in food, and when we talk about carbohydrate-rich foods, we mean the ones where there are polymers of glucose that can be broken down by the body into glucose, which is an energy source for all of the processes that go on in our bodies. This is typically foods like rices and potatoes and things have large amounts of digestible carbohydrates. And it's typically located in the seeds. But carbohydrates also play important structural roles, and in fact, they also make up the bulk of the material in wood. It's just that we can't digest that particular carbohydrate. They're also present on the surface of cells and are very important for the recognition of between cells and recognition of pathogens and our immune response. But the carbohydrates I've been working on are these ones that are contained in other digestible ones. And the reason for this is that when I moved to Denmark, I started working at the Carlsberg laboratory. And you probably realize that barley is not just a source of food, barley is a source of beer. So if you look closer at one of these grains of barley and slice it open, it contains the major species inside is called starch. I think it's stilse in Danish. And this is basically polymers of glucose. Here's the structure of glucose, and here I represent it as a green hexagon, just for simplicity. So the glucose that we can digest is contained inside polymers, which are, there's two different types. There's amylose, which is basically a linear polymer, and amylopectin, which is a branched polymer but has these long branches. And I'm a supermolecular chemist, so I'm interested in how molecules fold up and how they interact with each other. So I got really interested in this when I realized that if you take one of these chains of glucoses and add a hydrophobic guest, something that doesn't like water, then the glucose chain will actually wrap around that molecule and form a helix. Now, this is not so different from the receptors I showed you earlier. These were cyclic species made from glucose called cyclodextrins. They have a hydrophobic exterior, hydrophilic exterior, and what we're seeing when you have a linear chain of glucose is that it's basically just like a stack of these cyclic ones as it wraps around. And the driving force for the interaction is the same thing. It's the molecules trying to get away from water because they don't dissolve very well in water. So one of the problems that we had at Carlsberg one of the challenges was to try to understand the relationship between the structure of the polymers in starch and their physical-chemical properties. Because when you make beer, you take the grains of barley and you heat them up in water and they swell, and enzymes start to act which break the polymer down into individual glucose, and then the yeast turns the glucose into ethanol. Now, if you want to do this process efficiently, which of course is Carlsberg's aim, to make as much ethanol as you can from a certain amount of starch, then you need to try and understand what it looks like and why there's variations from one year to the next. So my contribution to this was to take these types of molecules, just like the one I've actually been talking about, this Anthro File, and to get it to bind inside the branches of the amylopectin. And here, the goal was to try to correlate how it bound with the length of the branches and how many branches there were. This led to studies where we looked more closely at this particular interaction. So this interaction between linear oligomers of glucose and hydrophobic guests. And I looked at mixtures of these compounds and compared the different lengths of sugars and the binding strengths to different hydrophobic guests. So in this graph here, you can see that the length, the degree of polymerization, that's the number of sugars you have in the chain. And this is a measure of the binding strength, how well they stick together. And here we had two different Anthro Files, one with a longer tail and one with a shorter tail. But in both cases, you get a linear correlation between how many sugars you have and how strong the interaction is. So what this means is if you added one more sugar, you get slightly better interaction, slightly more binding. And what became apparent is that the strength of this binding is actually starting to get to the level where it could be useful for building materials. And that's where where this background at Carlsberg leads to what I'm doing now in the area of stimuli-responsive-materials. Because the question I'm asking now is, can we exploit this interaction to build materials based on glucose oligomers and binding to hydrophobic molecules? So what's the motivation for working with these types of materials? Well, there's various things. They're flexible, which is different to the cyclic structures that I showed, which gives us various possibilities in terms of what you can bind inside. They're based on natural polymers. They have the potential to be biocompatible, the potential to be biodegradable. And because they're made from glucoses that are stuck together, then they respond to enzymes. So you can add enzymes to make them longer and add enzymes to make them shorter. So that makes it a system we can control with enzymes. And then from a basic fundamental chemistry sort of level, this gives us an opportunity to learn more about hydrophobic interactions, more about specifically how these molecules form helices and also new approaches to making oligosaccharides. So broadly speaking, the two aims I have is, one is to try to make self-healing polymers. So these are polymers which, when you break them, they reassemble to form the original structure. And the way this would work is that we'd take a scaffold molecule and attach two of these glucose chains. Take another one and attach two hydrophobic pieces. And these, if you add these together, in theory, they might form a polymeric structure like this. But they're not stuck together with covalent strong bonds. They're stuck together with relatively weak bonds, which means that if they break apart, given time, they'll come back together again and reassemble. They're also potentially responsive to enzymes, because the enzymes can be used to break off each of these sugars individually or increase the length of the sugar chains. So then we have adaptive, self-healing, enzyme-responsive polymers. And the last aim is to look at cyclic, what are called cyclodextrins, these cyclic rings of glucose. Currently, these are actually used already quite often in the food industry, for example, to trap aromas and flavor molecules, and to stabilize these species, also to remove cholesterol from foods. But there's only 3 that are commercially available in reasonable quantities, where you have 6 sugars, 7 sugars, and 8 sugars. And because they're quite rigid, that affects what you can trap inside. You're restricted by the size of the molecules. But turns out these are made from using enzymes that take amylose, which was the material in the barley grains, and they spontaneously form these 3 species. So my idea is that if we take this system and add in a template that binds to a larger cyclodextrin, then maybe it will self-assemble around the larger species. And then we'll have access to a whole new series of molecules, which would be these larger cyclodextrins, which have not been studied to any great degree because they're difficult to access, but have the potential to be exciting new delivery encapsulation molecules. So that brings me to the end of the science I wanted to talk about today, and I'd just like to thank some of my collaborators on the recent work, Michael Pitterko and Sebastian Meyer. I've just started at DTU and have a very small group who are working on some of these projects now. So that's Vanessa Bay and Gustav Scheer Jansen. Thank you to Carlsberg Foundation, who are giving me my primary funding at the moment, also the European Commission, and of course UNESCO L'Oreal for Women in Science Prize. And then I'd like to mention some of the supervisors and advisors I've had throughout my career so far. So firstly, there's Barbara Mesley, who was my honors supervisor and gave me my first introduction to what research really is. Then Jeremy Sanders was my PhD advisor in Cambridge, and he taught me the importance of trying to look at new concepts, not just new molecules, but new ways of thinking. And I think that's good advice when you're trying to set out on a research career. And then Ole Hinsgaard was my advisor at Carlsberg Laboratory, and he's given me a lot of support and encouragement to pursue my own ideas. And so I'd just like to thank the 3 of them who've contributed a lot to my career thus far. And thank you to you all for coming.